Now, let's zoom in on the first half of our process. So first, creating our stable transgenic strains. So again, this is done by microinjection of the plasmid into Drosophila embryos. And it's at this step, at the embryonic stage, that the gene becomes stably integrated into the genome of the Drosophila. And as you can imagine, this is done with a really, really small needle, which sounds really challenging, but it's actually a process that's been done for decades. It's relatively straightforward, and there's actually a remarkable amount of control in this step. So we know exactly where to insert the gene in the Drosophila genome. So that is, we can actually target integration of the gene of interest into very specific predetermined hotspots in the genome where we know that expression will be optimal. Now, how do we know that the gene actually gets integrated in this step? Well, we do this by pairing our gene of interest to visual markers like genes that control for eye color. So we can actually select for flies where we know integration is successful. So basically, we can inject into embryos from white-eyed flies, and we screen for red-eyed flies in the resulting progeny. And so you can see in the middle image here just how easy it is to distinguish between those two populations. And so these are very specific advantages of the Drosophila system. Now, important to note, everything I'm talking about, everything I'm describing in this process till now is a one-time thing. So generating a stable expression strain only needs to be done once. So microinjection is a one-time thing. So once we have those stable strains, we can effectively rear them in perpetuity. And we can monitor this over time. We can monitor the genetic stability of our strains by utilizing these visual phenotypes. Again, we know in our process that with these strains, the red eye color is linked to insertion of our gene of interest. So in our expression strains, as we're rearing them, we can monitor for any occurrence of white eyes, which would suggest a contaminated stock, and we can rapidly correct that. So now, these stable strains are then scaled up in our novel rearing system. So scale up in traditional biomanufacturing systems is often fraught with challenges and very significant costs. So in those systems, once a master cell bank is established, those strains must be continually optimized over and over again in these subsequently larger vessels, which creates a really fragile process and that's prone to failure. And so because of that, scale up of a process for a given product can in itself take months or even years for traditional systems. In contrast, the EntoEngine scaling process is simple and robust, and that's what we designed it for. First, again, there's really high fecundity in Drosophila. A single female Drosophila can lay up to 100 eggs per day. So that allows us to exponentially increase populations. Basically, we can increase a given population 2 orders of magnitude every day. Contrast that with cell systems, which at best have 8 to 12-hour doubling times. Most CHO systems have a full 24-hour doubling time, while CHO-K1 will double their population in 24 hours. Drosophila are effectively 100xing their population in 24 hours. So it's really remarkable just how good insects and Drosophila in particular are at making more of themselves. This fecundity is further accelerated with our novel rearing process. So once a given strain is established, it's scaled up from vials to jars to trays over the course of a couple of weeks. Once a population reaches these trays, they can be readily and easily maintained at the scale, basically in perpetuity. So with these trays, we can rapidly scale up or down as needed. Production is very flexible, and that's because it's a modular system, which means we maintain smaller, discrete populations rather than one very large population. So that gives us more control over the process, but it has other advantages. It actually prevents the spread of disease. And so with that, our insect team takes really great care to keep these populations healthy and happy. The feedstock is prepared daily under near sterile conditions, and populations are constantly monitored for any sign of illness. The way our rearing process works is each population is reared for 24 hours on fresh food, which is poured into the bottom of each tray. You can see the yellowish looking material in each tray in the image there. And so that feedstock is a proprietary blend of ours, but it contains agarose, which makes it into a firm gelatinous-like substance. The adults then eat, mate, reproduce in those trays over the course of a 24-hour period. So the females will lay their eggs directly into the food. And after 24 hours, the food is basically saturated with eggs. And this is something that our team determined empirically and have optimized for the right population size for a given surface area of those trays. And so at 24 hours, those adults are removed from the tray and then given a new tray with fresh feedstock. And it's not because they consume it all, but again, because they saturate that surface area. Remember that females will lay up to 100 eggs per day. Once those trays have been conditioned with the eggs, they're moved into a separate chamber to hatch into larvae, where they then spend the next 3 days basically eating nonstop. They consume the entirety of the food and turn it into biomass. So at the end of day 4, they look something like the tray on the bottom, where basically the entirety of the feedstock is consumed and it's turned into larvae. And it's at that point that they are harvested, which is simply done with water and a mesh screen. And so this whole cycle of seeding trays and maintaining these populations is actually remarkably simple and easy to do, which was always by design. Because this process is cyclical on a 24-hour cycle, it allows us to harvest and therefore manufacture continuously. And this is a feat that's really unparalleled in the biomanufacturing industry. It is truly continuous production. We're the only system in the world, as far as we're aware, that can do this continuously, daily, in perpetuity with minimal resources. So once the larvae are harvested, they're stored in a frozen state. And depending on the protein of interest, this can actually be done for months at a time. We've been able to build up an inventory of frozen biomass that's been stored for months, and then we can still take that, process it, and purify fully functioning protein from it. And with our process, it's really easy to track every batch of biomass to the exact population. So we have full traceability in our process. And this is particularly important to note. We're not operating with GMP standards at the moment, but we fully intend to in the near future. And this aspect, the ability to trace our starting material back to each specific lot, is a really important part of enabling GMP standards. So how do we go from frozen larvae to purified protein? The next step is processing. Essentially, the biomass that's frozen is thawed. We run it through a mechanical homogenization step. This disrupts the tissue and creates a lysate. Now, importantly here is that our process doesn't require harsh detergents or chemicals to lyse the cells, which is often used in other expression systems. And that's because most often the protein of interest can be extracted directly from this raw homogenized lysate, which means we can employ gentler methods that don't disrupt or unfold the protein. We're not dealing with things like inclusion bodies. And that ultimately leads to a better functioning protein. After mechanical homogenization, the lysates are readily clarified with centrifugation, filtration steps to develop this clarified, transparent looking liquid. You can see in the middle, looks a lot like apple juice. So you can see just how clean we can get the lysate from a few simple steps. But just because the steps are simple, this process was not so simple to develop. Our team painstakingly developed some really innovative methods to make this process extremely efficient. Because of how efficient it is, we can already process tens of kilograms of biomass per day at just a benchtop scale right now. From there, everything downstream is more or less the same as other expression systems. So we utilize column chromatography for purification, plus some other steps like TFF to concentrate the final product, followed by lyophilization for storage. And again, importantly, is we can achieve ultra-high purity with our process. The EntoEngine is novel, but it's still compatible with all the downstream off-the-shelf solutions for protein biochemistry and purification.